So yeah, I'm your host, Dennis Koester, and this is the last coffee chat of the year, and we're going to be talking about the team's favorite molecules of 2024. So that's not necessarily our molecule of the year contest. You obviously still get to vote for those. Those are just our personal favorites that we want to share. And of course, this is like picking your favorite child. There's so many exciting things going on in drug discovery today that it's really, really hard to pick. And all of us had multiple favorites, and we had to work really hard to pick the one, if you will. Dive into the individual molecules. I wanted to briefly introduce what Drug Hunter actually is and what we do. Again, many of you probably know, but yeah, it's a really exciting time to be alive today in small molecule drug discovery, particularly. But it's also a challenging time, A lot of we get to hear a lot of content. There's a lot of buzz and noise. There's investors have expectations. You get company reports. You get conferences that happen every day. Every Thursday, the patents drop. So, there's literature that you need to keep track of. There's a lot of things that you need to be aware of in drug discovery to stay at the forefront. So Drug Hunter really is here to help you filtering that noise and get you to that signal really fast so that you can, do the things that, matter to you and help advance those drug discovery projects that you work on. So what we do is we gather from all of those sources that, again, are coming at you and are humanly impossible to monitor. We gather all those sources, monitor those, and condense this into like variety of different formats that are very digestible to you. So we have patent highlights, for example, where we distill monthly all the patent literature that comes out. We have case studies, for example, where we do analyses of a specific compound, where we go really end to end throughout the entire drug discovery story. We share a little bit about why this compound is important, in what context does it come, what is the competitive landscape maybe around the target that it's targeting, and we provide all of that in a report that is very easily readable for you and has also all these sources linked back so that if you wanted to dive into this a little bit deeper, you can use Drug Hunter as a jumping off board, if you will. Again, we have a variety of different tools that you can use as well to do your research. So we have a structure search tool as well. We have a text search tool. And you can easily search specific targets or indications. You can search for indications and find different targets and compounds for that indications that are operative. And you find things that are sometimes one-offs. Drug discovery is somewhat still largely empirical. Oftentimes there are ends of one, which is very hard to get data around, but we report those things that are really important, like a specific toxicity that has been observed, say an on-target toxicity or an unexpected off-target toxicity. Those are really difficult to find, but I would encourage you to try the Drug Hunter search engine, search for muscle wasting tox, search for kidney tox, something like that, and you will find case studies where people have observed that. And if you're experiencing something like that on your own program, it also has strategies how to overcome these particular issues. And yeah, Drug Hunter currently is trusted by large pharma organizations, biotechs, investors, and nonprofit organizations. We've have over 180 members of Drug Hunter as of today, and it's rapidly growing. Early career scientists trust us for accelerating their learning. Really, when people join from a synthetic lab, it takes some time to get familiar with medicinal chemistry, and Drug Hunter provides a nice platform to accelerate the learning. But also for scientists that are more experienced in their career, it helps you to stay on top of the literature, on top of the patents, and on top of everything that's coming at you, so you can focus on stirring that portfolio that you have in the right direction. We often hear from our members that Drug Hunter is one of the reasons they are actually able to stay on top of medicinal chemistry and drug discovery in general. So with that, I'll hand it off to our CEO, Dennis Hu, and he'll talk about his favorite compound of the year. Dennis, stage is yours.
All right. Thanks, Dennis. I had to change my molecule actually because I think there were just so many interesting ones. So I think this molecule from Revolution Medicines, this KRAS G12D on inhibitor that acts as a tricomplex molecular glue, is absolutely fascinating. Remarkable mechanism of action. So the way it works is it forms an intracellular complex with a cytosolic protein, cyclophilin A, and leverages that to induce neoprotein-protein interactions with KRAS, leading to the inhibition of KRAS. Obviously, for folks who have followed oncology or the KRAS field in particular, many people, had said KRAS was undruggable. Then people thought after, the drugs for the KRAS-C, G12C mutations came out that, well, that's nice. KRAS G12C is a minor mutation on KRAS, but, you're unlikely to be able to find covalent inhibitors for KRAS G12D. And now Revolution has a molecule in phase 1 with initial signs of bioavailability and safety that does covalently target KRAS G12D. So I just think this is fascinating for a number of different ways. One, truly a nice example of drugging the undruggable. But from a fundamental, chemistry perspective, there really aren't that many great examples of clinical candidates targeting aspartate residues or carboxylic acid residues. So a beautiful example using a novel aziridine warhead to be able to tackle that. So I'm sure we'll see aziridines pop up all over the place now in covalent libraries and the like. But you look at this molecule, this beyond rule of 5 thing, and it's just a truly impressive piece of chemistry. On a more personal note, I think some people may know that my wife is currently being treated for advanced colon cancer. She's actually starting chemo in a couple weeks. And I think we sometimes maybe lose sight of this, just being in the weeds of drug discovery, but patients are truly waiting for new medicines. And when we're able to tackle new targets for the first time, driver mutations like KRAS, even if people are not being treated with these things immediately, right, it gives people a lot of hope to know that in the future, there may be a drug for, your disease, or if your cancer comes back, what is there for them? We actually got the pathology or the molecular diagnostics panel from her tumor about 2 weeks ago, her primary tumor 2 weeks ago. And actually, I was really shocked to see that she has a KRAS G12D mutation. So she's, obviously very young. This is very rare in people of her age. But, it's just kind of flabbergasted to see that, her tumor is not highly mutated, and it has basically one, major action, what is now an actionable mutation, KRAS G12D. So having worked on KRAS G12C myself and, seen people working on, G12D, it's, really, again, flooring. So truly impressive chemistry, fantastic work, and just a testament to how important our field is.
Awesome. Thank you, Dennis. And I'll hand it off to Matt to talk about another very interesting compound as well.
So I promise you this is not being sponsored by Revolution Medicines, but yeah, my favorite molecule of the year is easily the pan-KRAS. Actually, it's in fact pan-RAS acting tricomplex glue inhibitor from Revolution Medicines. And as Dennis said, KRAS was this like archetypal undruggable target that everyone wants to go at, and it was G12C is it's a crack in the door, and this is really kicking the door in. This is really active. It's active against a broad swathe of mutants. And if you look at the crystal structure down here, you can see really why it's active against all of these different mutants, because actually the binding interface between cyclophilin and KRAS is away from these hotspots where these mutants happen. And actually that interface is conserved not just in KRAS, but in NRAS and HRAS. So this compound actually inhibits wild type KRAS as well. And what Revolution Medicines have seen is actually this goes against the kind of received wisdom in the clinic looking at G12C specific compounds, that it was thought that actually wild type activity led to a lot of the off-target toxicities, but really this compound looks a lot cleaner, and it actually deals with a lot of the kind of escape mechanisms that you see with G12C targeting compounds, where actually what happens is you have upregulation of wild types to counteract it, but the fact that you're actually getting wild type inhibition as well really is what's driving a lot of the activity that you see here. Again, it's a beautiful looking molecule. I think chemists are drawn to compounds that really look wild like this. You know, it's a macrocycle. It's got a hydrazine in it. It's got a lactone. It's atropisomeric. But still, they've managed to make this on scale. And this is beyond rule of five, but this is actually CNS penetrant as well. And they've shown activity in gliomas preclinically. And so I think this is just a really, really impressive molecule. So actually, Revolution Medicines had an update on the clinical trials a couple of weeks ago, and this is moving forward in phase 3 for pancreatic cancer and for non-small cell lung cancer. And it's really just wild activity they're seeing. I mean, it kind of recalls some of the early clinical results that people saw with PD-1 axis modulators, right? People who are really sick and are just being pulled out of the grave to a certain extent. So this for me is kind of a triumph of both chemistry, of pharmacology, and of thinking, well, if people say it's not druggable, they're just not taking the right approach. So this, for me, really is my molecule of the year.
Awesome. Yeah, thank you, Matt. Thanks for sharing this. And for those interested, we actually have a flash talk coming up by Adrian Gill, who's the basically SVP of chemistry at Revolution Medicines on January 23rd next year. So we would encourage everybody to tune in for more details on those stories. And again, feel free to read all those stories on Drug Hunter as well. And with that, I'll hand it over to Brett, and we'll hear more about his favorite molecule of the year. Oh, you're on mute, Brett. Sorry.
Mute me. Okay. Can everybody see me?
Yes.
Yeah.
Can you see me? Okay. So, hi, everybody. My name is Brett Buchser. I'm a new member of the team. And I want to talk about my favorite molecule, which is this molecule with the funny code name AZPRMT5I-1. And what it is it's what's known as an MTA cooperative PRMT5 inhibitor. The MTA cooperative aspect of it is that PRMT5 is an enzyme which is involved with histone methylation of arginines and histones, which is a cancer driver. And the MTA part is a co-binder in the PRMT5 protein, which takes the place of the cofactor SAM. And early on, there was a lot of excitement of discovery of inhibitors of PRMT5 to treat cancer but what happens when you inhibit wild type PRMT5 with inhibitors, you get hematological toxicities. And what people thought was, well, hey, we have this situation where there are 10 to 15% of people who have cancer have MTAP deleted cancers, which MTAP is this enzyme which metabolizes MTA. And these MTAP deleted cancers are specifically in cancer. And so if we can target MTAP deleted cancers specifically, we'll gain 10 to 15% of the cancer population. And the reason you want to do that is because in the MTAP deleted cancers, there's this molecule MTA, which is methylthioadenosine, and methylthioadenosine is also a weak inhibitor of PRMT5. And what companies began to do with second generation PRMT5 inhibitors is target the PRMT5 that's also ligating MTA, and the inhibitor takes the place of the arginine that normally goes into the binding site. You can see in the slide an image of the inhibitor in white or light gray and the MTA in purple, and you can see they're very close together. So a couple of things about the molecule which make it cool is the spirocycle, which they discovered later on to fix the binding conformation. The overall molecule is fixed in a binding conformation with that spirocycle. Then there's this NH2 group on the pyrrolopyridine, which takes the place of the arginine NH2. And that NH2 is key to potency. And you can see in the image of the binding interaction, all these hydrogen bonds that the NH2 is involved with. And then the last thing about the molecule, which makes it really cool, is what I'm calling a fantastic fluorine atom. It's right next to, it's vicinal to the NH2 group. And the reason it's fantastic is because it does more than just block a metabolic soft spot. That fluorine is also involved with the hydrogen bonding interaction with the vicinal NH2. That hydrogen bonding interaction makes the molecule less polar. You can see that reflected in the table, in the clearance numbers for the in vitro clearance, and also the PK, the in vivo clearance numbers. They decrease dramatically. Also, you get a dramatic improvement in permeability. So the Keiko-2 numbers also improve dramatically. The fluorine is probably also inductively changing the metabolic propensity of that NH2 group by drawing electron density out of it. So it makes it harder to be, it's more difficult to oxidize it with metabolic enzymes. So it also increases the half-life of the molecule. The fluorine is also probably withdrawing electron density from all that part of the molecule involved with hydrogen bonds to the Glu 444 and the Glu 435. And also very interesting in the structure image is this NH2 interaction with the S-methyl group of the MTA, indicating, you know, true like pictorial image of what is really going on in this binding complex. So I found this to be a very interesting subject. This is a very competitive target right now. There are 3 companies, actually 5 companies are in active clinical trials with these types of inhibitors. Amgen, Mirati, and Tango, and AstraZeneca, 4, and then 6, sorry. And then in the last 3 weeks, Tango, IDEA, and BeiGene all announced they're in clinical trials with PRMT5 inhibitors. So it's an exciting target to be involved with. And I thought that this molecule was very special because it can teach us things about medicinal chemistry that go beyond, of course, this anti-cancer approach. So there you go.
Yeah, thank you, Brett. That I found this target incredibly interesting as well. So yeah, thanks for sharing that story with us. All right, we'll hand it off to Shaima. Shaima.
Yeah, thank you, Dennis. Hello, everyone. Today I'll be discussing one of my favorite molecules of 2024, which is RLY-2608, which is a next generation PI3K alpha inhibitor developed by Relay Therapeutics. So RLY-2608 is an oral wild-type sparing allosteric inhibitor targeting PI3K alpha, a protein that is mutated in over 30% of breast cancer patients. So if you look at this figure, the first generation PI3K alpha orthosteric inhibitor alpelisib, and the recently approved PI3K alpha degrader inavolisib, they have shown clinical benefits actually, and this is why they got the FDA approval. But they come with limitations, and these limitations are due to their activity on the wild type PI3K alpha. And you can see in the table that their selectivity is, or they almost are not selective for the wild type versus the mutant. And yeah, one of the most important toxicities is the hyperglycemia, in addition to other on-target toxicities like rash and diarrhea. So for hyperglycemia, the prevalence, or it occurred at a 64% rate in phase 3 trials for alpelisib and 59% for inavolisib. So this is pretty significant. So these toxicities highlight the need for a second generation PI3K alpha inhibitors, which hopefully mitigate these on-target toxicities. So here we see the second generation mutant PI3K alpha allosteric inhibitors, which are more selective to the mutant versus the wild type, and hopefully they're gonna be devoid of those toxicities. RLY2608 and STX478 both are allosteric inhibitors, and they are in phase 1 for STX is in phase 1/2 clinical trials. And by the way, both bind to the same allosteric binding site. For LOXO783, actually, that molecule was in clinical development, but recently it was discontinued in phase 1 clinical trials. However, Eli Lilly, they have another next generation PI3K alpha inhibitor in the pipeline, and the IND submission is expected to be submitted next year. So if you look at the selectivity, like those next generation molecules are at least 10-fold more selective for the mutant versus the wild type, and this is expected to reflect later on the in the clinical trials. So for the discovery of RLY2608, it was discovered through a DNA encoded library or DEL screening. And by the way, we have a very interesting article on Drug Hunter that like explains everything about DNA encoded library screening. This technology enables rapid exploration of chemical space. They definitely like did a lot of strategies during the screen, but one of the most interesting strategy was blocking the orthosteric binding site so they can hopefully identify allosteric inhibitors. And why is this? It's because the mutations in PI3K alpha mostly do not affect the orthosteric binding site. So, and this is why alpelisib, unfortunately, is not selective. The DEL screen was followed by subsequent structure-based drug design and optimization that led to the identification of RLY2608. So the selectivity of RLY2608 arises from structural differences, which leads to also different binding kinetics for the mutant versus the wild type. So when it comes to the structural differences, in the wild type PI3K alpha, the C-terminal tail, like it stabilizes a critical region and makes the allosteric site inaccessible. Or on the other side, when it comes to the mutant, that C-terminal tail is like more disordered, so that allosteric site is more accessible. And this reflects absolutely on the binding kinetics. So RLY2608 has a faster association rate and a slower dissociation rate for the mutant compared to the wild type. This leads to a longer drug target residence times on the mutant and hence more selectivity. RLY2608 in combination with fulvestrant is currently in phase 1 rediscover clinical trial. The interim data highlights the compound's clinical activity. The objective response rate was 39% overall, 67% in kinase domain mutations. The progression-free survival was 9.2 months overall and was 11.4 months as a second-line therapy after the CDK4/6 inhibitors. And there was really some differentiation in the progression-free survival versus other standard of care treatments. When it comes to the safety profile, that was the most exciting part. Grade 3 hyperglycemia occurred only in 3% of the patients, and diarrhea occurred in almost 2%, grade 3 diarrhea. So we see like a real improvement in the hyperglycemia in the mutant selective inhibitor RLY2608 versus the first generation inhibitors. So moving forward, the company is planning a phase 3 trial in 2025, and this is pretty exciting. And I think just RLY2608 represents a significant step forward in achieving mutant selectivity and sparing the patient all those very unpleasant side effects that makes it even like it might limit even the benefit of the of these drugs. So thank you for your attention, and I'd be happy to take any questions at the end of the session.
Yeah, Shaima, thank you for sharing this. And for those of you who don't know, Shaima also recently was at the San Antonio Breast Cancer Symposium and came back with a fantastic conference highlight summary. And some PI3K inhibitors are also mentioned in those particular conference highlights. So I'd really encourage you to check those out as well, and all the other mechanisms that are being used for breast cancer. So really kudos to you, Shaima. What a fantastic conference highlight summary.
Thank you, Dennis. And I want to add something about the conference, actually, just to add to Dennis' point. So there were absolutely many oncologists on that conference, and it was very inspiring that you hear oncologists talk about the drugs and the side effects and how, for example, they don't like to use certain category of drugs because of how hard it's for the patient to tolerate the side effects. So yeah, attending that conference was very inspiring for me.
Yeah, thank you for sharing that. And I think, Shaima, this goes to what Dennis alluded to in the beginning, right? Drug discovery is largely personal to all of us. And a lot of the times when people talk about their motivation as of why they started a career in drug discovery, it's because of a personal story. So I believe we're all touched by those personal stories and how drugs really affect patients. So that is definitely one aspect beyond the science that we do want to keep in mind. And with that, I will hand it off to Rory, who's also going to talk about a massive problem that exists in the US that maybe there's a better solution to that with this
Something on the
drug.
horizon. Yeah, thanks so much, Dennis, for organizing this. This is always a really fun topic at the end of the year to think about and to discuss with the community. And so one of my favorite molecules from 2024 is Vertex's non-opioid selective NaV1.8 inhibitor, VX548. The in name is suzetrigine. I can't pronounce it. Suzetrigine. And the reasons why I like this molecule so much are really sixfold. And so, Dennis and Shaima just touched upon this personal story aspect of drug discovery. And so that's, really one of the first reasons why I like, VX548. So there's a significant unmet need for non-addictive pain medications, given that over 21% of US adults experience chronic pain annually. And so that, if you think about those numbers, that's over 50 million people in the United States alone are experiencing chronic pain. And that doesn't even include the folks that are dealing with acute pain, after surgeries and procedures. And also, given the ongoing opioid epidemic, which has, some of the numbers are around 500,000 deaths have been attributed to the opioid epidemic. I mean, there's just a huge unmet need here to find alternatives to opioids. And so that, personal story, that personal aspect of VX548 is something I find very, very, very interesting and important. Another reason why I like this molecule so much is really the compelling story behind it. And what I think is so compelling about 548 story is that NaV1.8 wasn't always seen as the hottest NaV isoform to target for pain. And in fact, the NaV1.7, the closely related isoform, gained attention after the 2006 discovery that mutations in this channel could render humans less sensitive to pain. And so after this discovery in 2006, there was a huge onslaught from the industry to find and to target the NaV1.7 isoform. But surprisingly, NaV1.7 inhibitors have yet to show really meaningful clinical efficacy, despite this huge industry-wide hunt for novel analgesics in this space. And so I think it's really this perfect example of how difficult it can be to translate even the most compelling genetically validated targets into therapeutics. And so that's a really interesting part of the story that I find very fascinating. With regard to Vertex, Vertex's interest in the pain space, their journey to 548 has spanned over 2 decades, if not longer, starting with their acquisition of Aurora Biosciences in 2001. So Aurora is this biotech in San Diego. They had expertise in ion channel chemistry and high throughput screening platforms. They had expertise in the CFTR. Actually, within the facility walls of Aurora, that's actually where ivacaftor was discovered. And so this really laid the groundwork for Vertex's pain program after this acquisition. If we think about the timeline and the continuum to 548, there are 3 other NaV1.8 inhibitors that Vertex had progressed to into clinical trials, including VX-150, this phosphate prodrug that you see on the slides here. This progressed to phase 2, and because of the high pill burden, was never really advanced to phase 3. They also looked at VX-128 in the clinic, as well as VX-961. Those two structures haven't been publicly disclosed, but they probably fall within the framework of what 150 and 548 look like. They probably fall on that continuum there. But so essentially, this is the fourth shot on goal, at least that we know of publicly, of Vertex, making an effort on taking a molecule into the clinic for this pain indications, and I think this really highlights this perseverance required to meet this moment of finding alternative to opioids. A third reason why I like this molecule so much is its thoughtful design. And so if we look at 548 compared to 150, 548 is exquisitely selective. If we look at this selectivity profile, it's 31,000-fold selective over the other 8 NAV isoforms, and this, in my mind, is just remarkable. This level of selectivity. Also, the potency is dramatically improved over 150. So it's a sub-nanomolar compound. So this level of selectivity is not only amazing, but it's also important, as we all know, given the role of NAV 1.4 and muscle contractions, and NAV 1.5 as an off-target in regulating heart rhythms. And so finding that molecule with that exquisite selectivity, I just think is really, really impressive. A fourth reason why I like this molecule is it has an impressive clinical efficacy. So in phase 3 trials for moderate to severe acute pain following a bunionectomy and tummy tuck procedures, it offered pain relief comparable to opioids like Vicodin. And so if you think about Vicodin, it's a combo med of hydrocodone and acetaminophen, and you have a single molecule that has the comparable efficacy to a combo medicine, which is, in my mind, pretty impressive. And if we look at the safety profile, it has placebo-like safety, which is again awesome. No signs of addiction or anything like that. So phase 2 trials have also showed positive results in treating chronic painful diabetic peripheral neuropathy, which is encouraging. But as we all saw yesterday, there were some mixed results that came out yesterday, highlighting how really heterogeneous and challenging these pain indications are, especially for chronic diseases. And some of the confounding placebo analgesic and placebo effects that we see oftentimes in CNS drug discovery. And there's actually a really nice Nature article that was published in July 2024 that highlights this exact phenomenon recently from a team at UNC. They look into this placebo analgesic effect very recently, and so certainly these results just highlight how challenging of a area this is. Just 2 more reasons I wanted to touch upon is the fifth reason is really the chemical structure. And if we look at 548, it's really just a beautiful and stunning molecule. It has this highly decorated THF ring containing 4 contiguous stereocenters, including 1 which is quaternary. You can take a look at our article on 548 and look at the process, the manufacturing route, which I think is also equally impressive. And so take a look at the article for that. So the chemistry and the structure is also just amazing. And the last point I just wanted to touch upon is, this molecule has really ignited industry interest. Companies like Latigo Biosciences has raised over $135 million this past year in Series A funding. And just earlier this week, I think on Wednesday, Site One Therapeutics, they've been around for a while now, announced that they secured a $100 million funding round backed by Novo Holdings. And so, clearly there's this huge industry interest or reinvigoration that based on 548. And so really those are, I think, my 6 reasons why I really love this molecule and why I find it so fascinating. Just to touch upon Site One and Latigo, we do highlight these molecules in our coverage on Drug Hunter and talk about their differentiations from one another and things like that. So feel free to take a look at our articles to see how they're different from 548. So I think, maybe just some concluding thoughts here would be that we have an FDA decision that's expected really any day now for moderate to severe acute pain. And I think 548 stands as this testament to what's possible in drug discovery, if we have the perseverance to follow through. So that's my molecule. And thanks again, Dennis.
Yeah, awesome. Thank you so much for that, Rory. Yeah, really fascinating story on so many different fronts. So yeah, thank you for shedding light on all of these fronts. Really very, very helpful. All right, with that, I'll pass it on to Lew to talk about one of his favorite molecules of the year.
All right, thanks, Dennis. Yeah, I've spoken about this molecule a number of times over the course of this year, and it's definitely my favorite molecule of the year for 2024, and that's for a number of reasons. And some of those are sort of captured even in the title, right? A reversible covalent allosteric, an ATP cooperative inhibitor of Werner helicase from chemoproteomics. So this story I find interesting from soup to nuts, really from the selection of a previously considered to be undruggable target, to the hit finding approach that was used, to the mechanism of action, to the chemical structure, all the way to the disease indication. So really for end to end, I think there's just a lot of novelty and innovation here in this story. Really is exciting for all those reasons. So, Werner helicase, as folks know, belongs to a class of proteins that for many, many years was considered to be undruggable or at best difficult to drug. And so, this whole drug discovery effort falls into this area of synthetic lethality. This is an approach to oncology therapeutics that tries to leverage the dependence of certain tumor types on two basically proteins in particular. And so if you shut one of them down, then you basically create a situation where the cell simply can't survive and proliferate. And so this is sort of in that arena. So one thing that I loved about this story, like I said, is this chemoproteomics approach. So this approach to hit finding has gained a lot of interest over the last 5 to 10 years. A lot of great work has come out. And one thing that I find fascinating is that by using chemoproteomics and going after covalent inhibitors, they were able to ultimately come up with a molecule that is very, very small and yet very, very potent and very effective. And this is in contrast to some other molecules in this space that took a non-covalent approach. They found wonderful molecules, but those molecules eventually had to be engineered in such a way that they're beyond rule of 5. And that's a fascinating approach in and of itself. But I love the elegance of this molecule. So how does this molecule work? So they've elucidated the mechanism. It binds covalently to cysteine 727. And so one thing that they leveraged is this alpha beta unsaturated sulfone warhead. This has become quite common in the Werner helicase arena. I personally hadn't really seen much of this until Werner helicase inhibitors came to the fore. And one thing that they found during the lead generation phase of this program is their initial hit molecules were actually competitive with ATP. So these are allosteric inhibitors, but they bind to a site that's adjacent to the orthosteric site where ATP binds. And initially they were competitive, but they found as they were prosecuting their lead generation program, a few subtle structure modifications in that initial hit wound up giving them a molecule that showed higher activity in a whole cell assay than it did in more biochemical type assays. And they elucidated what was going on. And what they found is by engineering the molecule further to optimize its activity, they actually flipped a mechanism from being a competitive inhibitor to cooperative. So it works better in the presence of ATP. It binds alongside ATP. So this is good because in the physiological context, there are higher concentrations of ATP, and this actually serves to make this molecule more efficacious. And then beyond those sort of facets, this this molecule, if you look at it, you can see that it's well within the Lipinski rule of five sort of paradigm. And I think Dennis is going to be talking about a molecule, unless he switched his favorite molecule for the year. I don't remember. You're going to see quite a contrast between the two of these. So again, I've spoken about this molecule quite a bit. I think it's an elegant solution to a problem that was previously intractable. And you know, it's going to get my vote this year, and I hope it gets some other folks' votes as well. Thank
Awesome.
you.
Yeah, thank you. I did not change my molecule, which is it's good. Yeah, so that because you already did all the work for me, right? You already talked about why this is such an interesting target. And yeah, I want to highlight this discovery story here for HR0761, which is also an allosteric inhibitor of Werner helicase. But as Lew pointed out, this is a non-covalent compound. So there are three reasons I really like this campaign, and those I will highlight here throughout. And I think there are some learnings here for everyone to be had. So first, the first reason I'll talk a little bit about their hit finding approach, which was completely different from a chemoproteomics platform. So basically Novartis at that time threw the kitchen sink at it, everything, every hit finding method you can imagine. And it was very, very difficult to find hits. You know, there are a lot of false positives in this space. There are no approved drugs actually for either DNA or RNA helicases. And Werner helicase also has an exonuclease function, by the way. So yeah, really it was very, very difficult to identify hits. So they had to constantly refine the assays that they're using until they found a binding assay. Initially, they also found ATP competitive inhibitors. And then they identified a binding assay that was more sensitive as an ATP binding assay. And they really had to tune their assay cascade to make sure that they filter out these false positives hits. So they had to use things like SPR and NMR to make sure that there was some on-target binding. And really, I think that highlights how complicated sometimes drug discovery is. You really want to make sure that you have your assay cascade set up. And, oftentimes you also need a little bit of luck, because what happened is that they found and identified a singleton hit from that whole effort that they moved forward into the lead optimization. And yeah, we have a nice flash talk from Dean Brown also talking about how hit molecules look like compared to clinical candidates. And, this is one of the examples where the core structure looks, very simple, but it was grown a lot through very, very, I think, thoughtful lead optimization. And that was one of the points I found it interesting. And 2 points I want to highlight in the lead optimization that I found interesting. One point is the so-called chameleonic transformations. So I think one of the great challenges in the beyond rule of 5 space is to predict chameleonicity. And this is because you want your molecules to still be orally bioavailable, although they may be in a space where that is challenging. So for those macrocycles that we've seen as, PCSK9 inhibitors, KRAS inhibitors, and, the KRAS inhibitors that Matt and Dennis presented, it's really difficult to get those compounds to be orally bioavailable, right? And for that, it would be really great to be able to predict this chameleonic behavior. And what Novartis did here is they leveraged 2 really interesting chameleonic transformations. So, oftentimes they describe chameleonic transformations as red herrings, but those are really true, and they really investigated that very thoughtfully. And I want to take a moment to talk about those. So one was the introduction of this ortho substituent here in this anilinic amide, if you will. And what that led to, surprisingly, if you would typically introduce a chlorine or a methyl group, you would expect that the log D would tend to be, to go up. So the compound would be more lipophilic. But here, unexpectedly, the compound had a very similar log D when they measured it. And that actually had to do with the distribution of the conformers. So they, one conformer is kind of the, bioactive conformation, but they also were able to identify a minimum PSA conformer, which is also kind of equivalent to the most permeable one, and a minimum LOCD conformer, which is the most soluble one. So really, the introduction of this ortho substituent did a variety of different things. It influenced basically how the compound behaves in different environments. It acidified this NH here, but that acidification was counteracted by a steric shielding effect, so that the hydrogen bond donor moment was actually lower compared to the unsubstituted compound. So this ultimately led to still a compound that could permeate membranes, but in an aqueous environment was very soluble. So very interesting transformation. The other chameleonic transformation that occurred was here in this part of the molecule. And they originally identified as salicylamide, but now this is a picolinamide, as you can see. And there, they leveraged this interesting concept of non-classical Zwitter ions. So when you talk about or when you think about Zwitter ions, typically you think about that your acidic pKa needs to be lower than your basic pKa. So your acid is essentially deprotonated and protonates that base. But it's actually a fairly general phenomenon that non-classical Zwitter ions exist, where the basic pKa is lower than the acidic pKa. And this is one of those phenomena that's been described in the literature, that this phenomenon generally exists with those, picolinamides. So I would encourage everybody, who needs a transformation like this in the beyond rule of 5 space to try out this modification. So what the Zwitter ion actually does is very interesting. So that Zwitter ion only exists in the ionized form in water, right? So in that water, it would be soluble, but actually when it goes through the membrane in a hydrophobic environment, if you will, it exists as the neutral form and is still permeable. So what Novartis actually found was that, the LOCD of these compounds was 2 units off, and that's like 2 orders of magnitude. The compounds were 2 orders of magnitude more polar than expected. So, very, very surprising transformation. And it is a fairly general modification that I encourage people to leverage. As they were able to, and now that gets to my third and last point about this compound, which is the neutral TPSA. So what they wanted to optimize really, at the same time they wanted to optimize lipophilicity. They didn't want a too lipophilic molecule. They wanted, a high lip-E, but, that is driven by low lipophilicity. And at the same time, they needed really crucially a cell-permeable compound, because this is an intracellular target, and you really need to get into that cell and need to be permeable and absorbed, for that matter, to even have an effect on the target. So they leveraged this interesting concept here of neutral TPSA. So that's the 2D PSA minus the 3D PSA. And that's really a bell-shaped curve, if you will. So there's a sweet spot. And the sweet spot, they called this very interestingly and memorable, the rule of 1/5. If your neutral TPSA is in between 1/5 and 1/3 of the molecular weight, you are in a good spot of lipophilicity and permeability to be balanced. If you're a little higher than that, you're going to be too polar, and if you're a little lower than that, you're going to be too lipophilic. So, as a nice parameter to optimize your oral bioavailability and your permeability and lipophilicity at the same time, try using that neutral TPSA and let us know how that goes. And yeah, those are, there are a lot of other things in this molecule that are interesting, but those are the things that I did want to highlight. And I wanted to let everybody know all of these case studies can be found on Drug Hunter, and we have hundreds of more of case studies that Lew and his team are putting together, really on a very frequent basis. I want to thank all of you for sharing your favorite molecules with us, and we will open it up for audience questions. Before, I want to thank our entire team. You've seen our content team here today, but to be able to put this content in front of you requires really large work behind the scenes. And I want to thank everybody from the dev team, the operations team, the research team, really for their hard work to make this possible for us to be here and present those cases to you. And with that, we'd love to hear your questions and thoughts about molecules. And I'll open up to the audience. I haven't seen Q&A yet, but I'll stop sharing and then I'll look into the Q&A box.
So we've answered a couple of questions already. So one coming from Matt Fife was asking about Revolution Medicines, the Zerodine ring opening. And actually that was key in their lead optimization, balancing the kind of covalent modification of their target aspartate versus the chemical and plasma stability. I think they use simulated gastric fluid was their main thing driving the SAR around this. And actually it's adjacent to the cyclopropyl ring. It's where it opens up, and by changing the substitution on the nitrogen at this position, they're able to balance the activity with the stability of this compound. So it's actually a key point of the lead optimization.
Awesome. Yeah, thank you, Matt. I saw that. And then Rory, do you want to address Alice's question?
Yeah, so I definitely don't think it's the end of the road for VX548. You know, it's certainly not in acute pain. I believe along with the announcement yesterday, the company Vertex announced that they're planning on restructuring and rerunning their phase 2 trial for this chronic lower back pain indication. So yeah, I definitely don't think it's the end of the line for this molecule. It just goes to show, you know, how heterogeneous pain is, you know, depending on the indication, depending where the pain's at, chronic or acute. You know, companies have to be very you know, deliberate with how they design their clinical trials to test their hypothesis. So
yeah, thank you, Rory. And then Shaima, I see you're already typing an answer, but maybe you can answer for us. Arun is wondering, do you think a 12-fold selectivity against the wild type, and he's referring to the wild type PI3K alpha here, is good enough to achieve the desired safety margin for a Relay's compound?
Yeah, I just realized that I can't like answer them in text, so I just was typing my answer. But anyways, in my opinion, absolutely, I'm not on Relay's team. As a neutral person, in my opinion, 12x selectivity won't be enough to achieve like absolute selectivity. And this is why we still see some cases of hyperglycemia in clinical trials. So we absolutely would like to see it more than that. However, the percentage of patients suffering from grade 3 hyperglycemia is less than compared to the first generation. So I think this improvement also is good for the patient. So there is, I would say, some degree of selectivity here, but it's not absolute selectivity. Yeah.
Yeah, that makes sense. Yeah. And I guess, yeah, we will have to refer to the experts, and we'll have to wait for the clinical trials to tell us more about those results.
Exactly. Yeah.
Awesome. Yeah. And then Hervé, hopefully I'm not mispronouncing this, is asking, great discussion. Thank you so much. Any comments on today's announcement of the failure of Novo Nordisk follow-up for anti-obesity? Maybe I can briefly comment on that and then hand it off to the team. So I don't know if I would describe it as a failure. Again, that's a subjective somewhat description. So I think what you're referring to is this report of the CagriSema obesity trial that fell about 2 to 3% short of analyst expectations when it comes to the endpoint. I mean, it's an interesting approach. It's because it's now looking at semaglutide, which is a GLP-1 receptor agonist, and cagrilinide, which is a long-acting amylin derivative, and you know that is an interesting MOA, I guess, that we're watching. But you know, I think it's too early to make comments on that based on this initial observations. But I would love to hand it, you know, maybe hear the team's thought on this. If there are any additional thoughts, we can share those as well. Anybody want to comment on this?
Yeah, I have a couple of thoughts. So one thing to keep in mind is that as drug hunters, ultimately we're serving patients, not shareholders. Now we rely on shareholders to help fund our drug discovery and drug development efforts, but ultimately it's the patients we're looking at. So that's one thing to always keep in mind. The other thing to keep in mind is stock investors have a pretty poor track record on predicting the ultimate value of an approved drug. So like Dennis said, it's too soon to tell. We need more studies, more data, and ultimately we'll see if this therapeutic compound provides, a safety and effectiveness profile along with tolerability and convenience that helps patients. And, frankly, from my point of view, if this turns out to be a $500 million per year drug instead of a $10 billion per year drug, but those patients being served are noticing dramatic improvements in their well-being and health and lifestyle, then I think, that's a win for patients.
Yeah, and in terms of kind of the change in the stock price, Novo Nordisk went down, but Eli Lilly went up. And I think it's that the market expects there to be one winner to shake out of this. But we have no idea. We can't tell what that is until everything comes out through the clinical trials. I mean, it'll be interesting to see. We've got small molecule compound agonists of GLP-1 are coming through at the moment. What will happen to the stock prices of Novo and Eli Lilly if those are successful? And there was a great article that just came out in Science Translational Medicine comparing orforglipron with danuglipron, and it looks like perhaps orforglipron might have the edge kind of pharmacologically. But I think Lew's right in saying, actually, at the end of the day, it's kind of what is the patients that matter most, not the stock price.
Yeah, no, thank you. And then we have Emilio. Maybe that rounds it up. We have about 2 more minutes. We had a question about PROTACs. If those were mentioned earlier in as earlier part of the studies. And does anybody want to comment on that? Otherwise, I can.
Well, no, we didn't feature any heterobifunctional degraders in the first part of the session. No.
So maybe an offhand comment here. So Dennis actually was selecting between a PROTAC, as he mentioned initially, and RevMed's KRAS G12D compound. So yeah, we definitely had thought about PROTACs. PROTACs are definitely on our radar and on our minds, and so are molecular glue degraders. There's definitely a lot of research happening in this field. They're expanding beyond oncology as well. We actually have a seminar and an article on that Dennis is featuring in. So yeah, we're very excited to reaffirm this about the space, about potential approvals early next year, maybe in the androgen or estrogen receptor degrader space. And yeah, maybe I'll let Lew comment. He's really the team's expert here for this particular topic.
I mean, this just goes to show how much incredible small molecule drug discovery is going on, that with all of the advancements in heterobifunctional degraders, the team, and again, this is just the team's decision, our membership and other people who vote on Molecule of the Year will have a say on Molecule of the Year. It just goes to show you that it's so difficult to choose your favorite. So, yeah, I mean, everybody's got their different choices for their reasons, and ultimately we'll see how folks vote for the Molecule of the Year, and it may be that a heterobifunctional degrader becomes the drug hunter molecule of the year.
Definitely. And Lew, do you know if anybody in the industry is currently working on trivalent PROTACs?
Well, I mean, there are these reports coming out, but do I know if somebody's really serious about it? No, I don't know to what extent, people are working in this area intending to put something in development. A lot of times in this space, as you know, a lot of companies have IV heterobifunctional degraders as part of their pipeline. Everybody wants oral. And, with all this talk we have about the beyond rule of 5 space and heterobifunctional degraders fall in that space, never forget, Lipinski never said you can't get an orally bioavailable drug in beyond rule of 5 space. He said, 10% of the drugs at the time he did his analysis were orally bioavailable and violated the rule of 5. But what we know, and what I know from, dabbling in this area when I was at Chimera, you can engineer these molecules to be oral, but you're going to have a tougher time. It's going to be a lot harder, and you're going to have to be clever using things like chameleonicity, using things, like Dennis talked about, the neutral TPSA approaches, very clever approaches using the Werner helicase program from Novartis. So, yeah, we'll see. The field's continually evolving, and if there's a disease indication that merits, using one of this sort of trivalent heterobifunctional degrader, bifunctional degrader, trifunctional degrader, it'll happen. It's largely going to be driven by the need and the sort of therapeutic hypothesis and so forth.
Yeah. Yeah, no, thank you, Lew, for that perspective, and I think that rounds it up very well. So yeah, I want to thank all of our speakers today. Thank you for showing us your favorite molecule. I want to thank the community as well. Rory will lead our effort in collecting your votes for the molecule of the year, and we'll have 2 rounds, and Rory can speak to that a little bit more. We're really looking forward. We're wishing all of you happy holidays and a happy new year. Hopefully, you have some good time off with your families, and we will be back in 2025, and we'll be here with the Flash Talk from Adrian Gill on the RevMed platform, which, as you know, is a very, very exciting platform. And we highlighted a few molecules here today. So yeah, with that, I would like to close the session. Thank everyone for their engagement. Thank you for all of your questions. Keep them coming. And yeah, see you next year. Take care, everyone. Bye now.
Bye, Dennis. Take care.